So I myself, Ashok Kumar Dasani, I'm a M.Pharm graduate with 8 years of experience, and I'm a SAS certified clinical trial programmer, and I'm also a SAS certified base SAS programmer. Apart from that, I'm a R certified programmer and Python and AIML expert as well. So we are from VRIT Solutions, where we deliver and fine-tune students mainly for clinical SAS and other type of job-oriented programs. We will take this session today for let us understand what exactly the clinical SAS is all about and how we will be navigating, how we will be leveraging this technology within the clinical sector. So what exactly SAS is? It's a statistical analysis system. We also call it as statistical analysis software. Previously, it was called as statistical analysis software. But after a huge amount of components that been joined into the SAS, now it is called as a system. And it is a computer programming language as well. And it is also the name of the software as well. And it is developed by the SAS Institute, which is from the North Carolina State University in 1969. Just give me 1 minute. Sorry for that. So SAS is developed by the Statistical Institute, which is in North Carolina State University in US. So way back in 1960s, when US Government of Agriculture, they want to analyze the agriculture data, they gave this project to North Carolina State University, the statistical department in the North Carolina State University. The statisticians in that department, they have developed and they come up with this programming language. By that time we were already having a very sophisticated and very costly software at that time, which is S plus, and people were paying very huge amount of money for statistical analysis. Then SAS came up with an idea that why can't we just create a programming language which is going to give free hand to the data analyst to analyze the data according to their will and wish. If you consider Excel, you can use only whatever the components or whatever the tools that is given along with it. But the advantage of having a programming language is it's up to you. However you want to mold it, however you want to design it, you can do it. So with that idea, SAS has been designed and developed in North Carolina State University in 1969. And when it was incepted, it was really meant only for agriculture industry. But later on, because of its wide range of capabilities, it is widely used in wherever data comes. In all those places, SAS is widely used. You take agriculture, SAS is there. Banking sector, SAS is there. Life sciences, 100% of the life sciences companies, pharmaceutical, biopharmaceutical, or biotechnology companies, microbiology companies, vaccine therapies. In all of these life sciences, the data, if you want to analyze, SAS is the answer. You take public sector, manufacturing sector, telecommunications, even in the retail and consumer goods. Right now, we are using SAS, in a wide range. Apart from that, even in the governance, SAS is widely used. Healthcare sector, SAS is widely used because of its, very sophisticated and very good statistical tools, statistical power, and it is also comes up with very ease. What else SAS can be done? We can use it for data management. When it comes to data is money right now. The more data you have, then you worth millions of dollars. Even sometimes, for example, if you consider ChatGPT is also data. Even though it's an AI tool, that AI tool is backed up with data. And what is the worth of ChatGPT? It's more than $10 billion. It's all comes up with the data. So this data need to be managed in a very secure manner. Otherwise, it can go leak and it will drastically draw the company down. So we need very safe and secure systems where we have to give very ease access to that data. So SAS is the place where you can manage the data as well in a secure manner. Apart from that, it is not only managing the data, it can give you the power to analyze the data in more advanced and most advanced manner. You can also perform multivariate analysis where you can draw the conclusions within wide range of wide range of variables. For example, way back, if you consider like 10 or 15 years before, we are all aware of that if in the TV, in the weather report, if the if they announce, if the reporter says that tomorrow there will be rain, drastically we will be not having rain. That's quite opposite which happened before. When they expect the cyclone to hit the ground, like within 2 days, it may hit within one day also. Sometimes it may hit very late, because the way that we were analyzing the data at that point of time is different. But right now, with the statistical power and with the huge amount of data analysis, we consider a lot of variables into picture, and we are giving the insights in a most precise manner. Now you have the exact location where that storm is going to come, at what speed it is going to come, and what will be the rainfall, and what will be the wind speed. You have all the parameters right now, and we are combining all these parameters and we are giving the outside, right? So multivariate analysis is the place where you analyze the data taking multiple variables into consideration. Okay, demand and supply, these are two different, these are two directly proportional. When the demand increase, when the supply decrease, demand automatically increase. But we don't consider other factors, the need of the people. For example, coconuts in winter season, mostly we will not get it. Even though the supply is very less, you will not claim that the demand will be very high and let me increase the price. Who will be there in the winter to drink coconut water? Very few, right? So we have to take other things also into consideration. So multivariate analysis will give you that power, how each variable is contributing to the output. Business intelligence is also a place where majority of the industries are driven by right now. They are all driven by the business intelligence. You take starting from Microsoft till to the small company in your local, all of them are using somehow the business intelligence to drive the company towards the profits, right? Predictive analysis is the place where we widely predict the future outcomes based on the established data, right? For example, weather is a prediction, stock price is a prediction, the likeliness of a disease for a particular group of participants is a predictive analysis. If you go to any astronomer, they do also predict, right? And that prediction, it can go wrong because they don't put all the variables into picture. But if a person, he is very well aware of the data analysis, and he is very well aware of all the statistical methods, and if he is very well aware of how to use them, how to take the dependent variables, 95% his prediction will go correct. Because the way we are analyzing the data, you need to have very sophisticated tool. You can't do it in Excel, because Excel will not take all the variables into consideration. You can drastically make 5 or 10 variables into consideration. What if you have like 20, 200, 300 different type of variables into picture? How can you predict it, right? So in this place we need SAS for prediction, like predictive analysis as well. Even the criminal investigation, SAS is widely used. So in all of these domains, you take like data management, advanced analytics, multivariate analysis, business intelligence, predictive analysis, and criminal investigation, SAS is widely used in some form. You take even the big companies like Google, they use SAS. You take small companies, they use also SAS. Because sometimes, is the SAS is the only answer? The answer is no. SAS is not only the answer, but we do also have other tools like R is there, R programming language is there, Python programming language is there. But the way SAS gives the commercial aspects to the industry is entirely different from what these open source tools are giving. If you take R and Python, they are open source, but whereas SAS is commercial. So something is commercial will always be backed up with support. Industries will always have that support, and industries will get very what we call good packages, good solutions they will receive. But whereas in R and Python, you have to prepare your solution. It will not come with ready made. You cannot bring every solution in a ready made manner. Sometimes you have to try it, trial and error method, you have to use it. Sometimes it may work, sometimes it may not work. But SAS is not like that, okay? Because you are paying for it and you will be getting the results and the output in a better manner. And industries, they don't rely upon one software tool. They do take multiple software tools into consideration. They take R, they take SAS, they take Python, they take some Spotfire and Power BI. Other tools also they will take into consideration, and they check the reports which are generated by these different tools, are they going to be same or different? If they are same, yes, they will come to conclusion that yes, our reports are fine and we are ready to submit. Okay. So what SAS can do? When it comes to SAS can access the data from wide range of resources. This is what we call it as, data analysis job role. So as a data analyst, even a data scientist, they do the, not data scientist, data analyst mostly, they do the same thing. They access the data and they do all the processing and they finally export the reports. This is what exactly any data analysis person will be doing. So SAS has all of it. It has the ability to access the data from Excel. You bring the data from the databases, it can access it. You bring the data in the unstructured form, it can access it. It can convert into a structured data set. So once you access the data, you need to explore the data. Soon after you receive some, you visit a place, you will explore the place for what are the opportunities you have there, what are the sites that are site of attraction is there, and how is the situation. You will be just exploring it, like how you are exploring the question paper after you receive it. Similarly, once the data you have received, you have to explore. The way of data analyst exploring the data is like he will check for whether the data is missing, how much amount of data gone missing, and what are the outliers of that data, and what are the summary statistics where the data is lying, right? Whether it is centrally aligned or whether it is widely distributed or a lot of outliers are there or whether it is up and down. So they will check with the help of some statistical tools how the data is distributed. Once the data is explored, they will go for preparing the data. Not all the data comes in, very handy. The data will not come, as how you want it. It may come up with a lot of challenges. Sometimes you need to clean, sometimes you need to remove certain observations, sometimes you need to add some observations, sometimes you need to calculate some columns, right? So all of those things we will be doing in the preparing data. Once we prepare the data, it is now ready for analysis. What type of analysis? It can be summary analysis, it can be frequency analysis, it can be any type of descriptive analysis. Once the data is analyzed and the reports are generated, we submit it for the export, where we export based on the client requirement. It may be sometimes the client may require the documents or the reports in Excel. Sometimes he need it in a PDF. Sometimes he need it for a presentation, the PowerPoint presentation. So all of these places, they may need that data to be exported. Okay. So as a SAS programmer, what you will be doing, you will be performing these 6 tasks. You have to learn how to access the data, how to explore the data, how to prepare the data for the need of analysis, and how to analyze what type of analysis are possible and how to do that, what type of reports the company may require it and how to generate them, and what type of outputs are the deliverables you have to prepare. Okay. Now we will take some insights about the SAS so that you will be uh, comfortable in learning SAS. First thing is, when it comes to the ranking of SAS in the world, it is number 2 in terms of artificial intelligence. It is widely used for text analysis. Majority of the chat GPT kind of AI tools behind the scenes, when they are processing the data for feeding or training the AI tools, they need the data to be clean. They can't rely upon some open source softwares at that point of time. So they were using SAS as well. The thing what I'm talking is about in 2015, because the inception of AI and all those things started way before 2015. The machine learning, when the machine learning started, even before Chat GPT, GPT-1 was there, when the first GPT was there, and they were having it. I think the name of the Chat GPT before was something else different. It was not Chat GPT. It was something, some other thing. I think. Then it changes its name. So majority of the AI tools, they take image as the data, they take text as the data, they take numbers or the tables as the data, they take videos as the data, right? Different AI tools, they are meant for giving solution with a different output. So for all of those, when you want to prepare the data behind the scenes, SAS is widely used. Even as a artificial intelligence software, SAS is used, and it is number 1 in the market with a market share of 28%, giving like the competitors are like Microsoft Power BI, and the competitors are like R programming software, SAS, Python, and you take whatever Spotfire, all of this. They share remaining 72 percentage. SAS alone took the market share of 28 percentage, and SAS is widely used as a in the journals, the BMC journals. 42.6% of the data analysis for the health related research is done with the help of SAS itself. And 100% of the life sciences companies, they use SAS as the software tool for statistical analysis. Now, when it comes to the picture of uh, data analysis in clinical, okay, there are some properties of clinical data. When you take banking data, if you open your any banking app, what are the columns you are seeing? The transaction ID, the date when you transacted it, the time, the amount credited, the amount debited, and what is your final balance. You will be having few variables. You don't have very huge amount of columns. You have very less columns. Maximum is 10 columns. And you take insurance, same. What is your policy number? What is the claim amount, and what is the paid amount, and how much is to be paid? How many months it has to be paid? You have like 10 columns. You take any data, business data, or any type of, you take retail data. What is the date that you have purchased, and what are the components? What are the groceries you have purchased, and what is the time you have purchased, and what is the total amount you have purchased in each and every month? Well, that much amount of data will be there. But when it comes to clinical data comes up with few challenges. Not only clinical, even the medical data, all of it has few amount, some properties. The first property is volume. When it comes to clinical data, you can draw the insights from a huge volume. You will be not having very small volume of data. You will be having very huge volume. Then the second thing is variety of data. It comes with huge amount of variability. For example, if you go to a hospital, and you are, by the time you are coming out from the hospital after taking few tests and those things, at least 20 to 50 different type of variables will be collected. Let us consider first one. Soon after you enter the data, they will be asking your vitals, right? What is your body temperature? What is your blood pressure? What is your glucose reports, and all those stuff? They will be collecting their height, weight, body temperature, heart rate, pulse rate. They will consider all of them. So in the starting itself, you have almost all given 5 variables. And the second thing, after you went to hospital, after you went to the doctor will give you, like, he will ask you a few questions about your to diagnose the data, to diagnose you. He will ask you a few questions that may contribute again 5 important uh, data points. And once you start, like, if the the doctor, he want you to check some, he asks you to go for some blood reports, or maybe like x-ray or scanning or something, then again it will contribute a lot of data. For example, if you are going for a blood report, at least 20 different type of parameters you will be collecting: WBC count, RBC count, glucose levels, renal function test, liver function test, so on, so enzyme analysis. You take like 20 different type of values, you will be collecting it. If the diagnosis is very big, you may contribute up to 85 variables also. Now, at the end of the day, he will give you also the medication. So this is also data. See how many different type of data are there. Your vitals are there, your diagnosis are there, your lab reports are there, then again your medication reports are there. Each of it comes up with different variables. Now this again poses a lot of challenge in the clinical trials. Okay, and the third thing, we call it as velocity. Consider going to a hospital. How many patients a doctor might diagnose or visit them? Maybe like you take 100 participants, 100 subjects, 100 patients, a doctor will see them in a day. For each of them, in a single day, if they are getting like 100 different data points, what is the total amount of data we are getting in a single day? 10,000 different observations you are getting them. This is only from one hospital. What if you are collecting the data in a city, where a city has almost like 100 hospitals, for example? Now, what is the velocity of the data now? It increases one more 100, right? 100 times increased. Then what if you are doing it for 30 days? See how much amount of data we are getting. Velocity, the speed that clinical trial data comes is very fast. And we need more sophisticated software tools to analyze this data and bring the insights in the most powerful manner and most easiest manner without any error as well. Now, can I say that now SAS is very, the clinical trial data is large and clinical trial data is complex? Can I get yes from all of you? With these properties, can I say that the clinical trial data is large and complex? Can I get a yes from anyone? Or no, also no problem. Am I audible?
Yes, sir, you're audible.
Yeah.
Answers yes.
Yeah. With this wide range of properties, we can simply claim that the clinical trial data sets are very large and complex. Now, to handle these large and complex data sets, we need a very proficient tool, which is SAS. That's why we are not relying upon Power BI, we are not relying upon Excel, we are not relying upon some other tools for analyzing our clinical trial data. And the fourth important factor is the value, which is very important factor in the clinical trial data. If you go to a hospital, how much money you will be spending for collecting all these to collect these 85 data points? At least 3,000 to 5,000 rupees. You go to a hospital and you are giving a blood report, at least 2 to 3,000 you will be spending up. Imagine
Yeah.
how much money is spent for all of these people to collect that much money, to collect that much data. Crores. Now, is our data clinical trial data has also a value? Huge amount of
Yeah.
value, right? So to handle all of these things, we need to have a proficient tool, which is again SAS. You can't simply share the data in an Excel file. If anything goes wrong, no one can identify. If I change the data points in it, if for example the hemoglobin values of a particular clinical trial participants is like one person is 17 and other person are having less than 12, all of them are anemic patients. But to claim that my drug is working, I will keep all of them as 13, 14, 15, up to 17. Now I will claim that my data is good. All my participants are having good hemoglobin. There is no problem with my drug. If I change the data points, will you be able to identify from the Excel? Change is change. That's it. You can't even identify whether I have changed it or not. Right? So there is a huge amount of malpractice can be done with the help of Excel. But SAS will not allow that. SAS can never allow that. So with those aspects, let us move further. This is to just to give you that the data that we are handling is entirely different than the data which is the others are using. So SAS can handle large data sets and it can handle complex data sets. You can perform data cleaning. You can also validate, okay, whether the data is changed or not. You can easily validate with the help of SAS. You can generate outputs like tables, listings, figures, graphs, and reports. You can easily prepare it with the help of SAS software. You can apply various statistical methods. Not always mean, median, mode, or standard deviation will work in the clinical trial data. We need to have very sophisticated, like we have different type of box plots, Bayesian analysis, or Wilcoxon test, some type of very powerful statistical methods, calculating a p value, calculating a t value, a probability values. So for all of them, you can't use Excel, you can't use Power BI. You need to have very you know uh, powerful tool to apply these statistical methods, that too with validation. Okay, and SAS can comply with the regulatory standard. This thing makes SAS more reliable and more important in the clinical trial industry, which is it can comply with the regulatory standards. At the end of the day, you have to submit the collected data to the regulatory authorities like FDA. This FDA, it has some requirements, regulatory standards are there. To meet out these regulatory standards, only SAS can fulfill it. Nothing more than that. The minimum application fees that you are paying for the reviewing of your clinical trial data is $200 million. Someone will not play around with this $200 million by taking Excel software, and they will also not take some participants or the subjects, from without having any good background. So they need good programmers, knowledgeable programmers, and logical programmers, and who feel responsible about the data, and who feel that who can apply the powerful statistical tools as well, right? So with all of these factors, SAS is number one in the clinical trial data analysis since 30 years, and it will be there in future as well. Now you might be listening a lot of gossips that SAS will be replaced, SAS will be having no job roles. That is bogus. That will never happen. Why? When it comes to clinical trial data, you just imagine uh, going from one city to another city. For example, if your salary is 1 lakh, okay, you want to travel from Hyderabad to Bangalore, and bus ticket is 3,000 rupees, okay, and train ticket you can take it like 500 rupees to 1,000 rupees, maximum is 1,000 rupees, for example. A flight ticket is going to be 5,000 rupees. Now which of these you will opt? Bus ticket, or train ticket, or flight ticket, when your salary is 1 lakh? Just give your inputs, just think of yourself. If time is an important factor, I will choose flight. If the business that I'm going to make in Hyderabad or in Bangalore, if it worth 10 lakhs, will I go in train or will I go in bus? I will not do that. I'll go by flight, which I can spend less time, and I'll be very confident, and I can deliver the speech there in the project meeting as well, and the project of 10 lakhs worth I will be receiving it. I'll be not playing any cheap tricks here. Sleeping in the bus and having neck pain, having body pains, and having dizziness in the morning and not able to speak in the project meeting, that won't happen, right? We will not. We are not ready to spare that much amount of time, money, and the value of the project. Similarly, here also in the clinical trial sector, the data that you are submitting to the FDA, it matters, billions of dollars in the pharmaceutical market. When you are bringing a sophisticated, bringing a new molecule into the market, which is going to bring $100 billion of value, you will not even think of spending $1 million or $2 million in purchasing the software, in hiring the people, in doing it. Because if something goes wrong here by using the free open source software, this $100 billion is gone, completely gone. So now will the company will take any uh, lenience in it? They will never. But is SaaS only going to be there in the market? The answer is no. Again, it will be working alongside of other tools as well, along with R also. Okay, that is the reason. That is the picture that you have to keep in mind. SaaS will not be replaced, but it will be working along with R. So people who know both of this, they can be working in the market for long term. Okay, so don't have any such type of conspiracies. So I'll quick I will take like 5 minutes to just give you the overview of what exactly the regulatory submissions are happening. To be precise, now we have understand what is the power of software, what is the power of SAS, and in what departments SAS is widely used, and what are the properties of the clinical trial data, and what is the amount of market, what is the size of the market that we had in the pharmaceutical market as well, pharmaceutical uh, sector. And now let us understand who are the boss for these pharmaceutical companies and why they rely upon SAS. So first thing is, in the clinical trial, whenever someone comes up with a new drug, if he claims that this particular new drug has the efficacy and it has the ability to cure a disease, then they will file first IND approval, which is like investigational new drug approval. So once they file this approval, once they got this approval, they will start the clinical trial. This approval will be given to the local IRB, which is like institutional review board, or animal ethical committees or ethical committee organizations will be there, where they will apply the rule, and even sometimes they will apply to the regulatory authorities itself that so and so drug is there with me and I want to start clinical trial in the humans. Now once they got approval, what they do, they go for the clinical trial. Once the clinical trial is started, they will started recruiting the participants and they will administer the drug to the participant, your clinical trial drug. Once the drug is administered, you have to collect the data from them, how they are feeling, what type of adverse events they are having, or whether the drug is effectively working or not. If it is working, what happened to the parameters, whether the glucose level is reducing or whether it is increasing, how much it is reducing, how many doses are required, what is the minimum effective concentration, what is the maximum therapeutic range. So all of these data components they will be collecting. So this data which we collect from the participant, we call it as raw data. Now this raw data will not be accepted by the regulatory authorities. They will not accept it because the way how you have collected is not standardized. So way back in 20-30 years before, the data which was collected, it was finely curated and they were submitting it. For example, GSK is conducting a diabetic study and Pfizer is also conducting a diabetic study, and take Roche is also conducting a diabetic study. Now the data that comes from GSK, Pfizer and Roche, it go differently to the FDA, which makes lot of time for the regulatory personnels to review the data and to give the approval for it. Now, to avoid all of these confusions, they come up with a new methodology called standardization of the clinical trial data, which is taken up by the CDISC organization. So this organization is a non-profitable organization where they take the responsibility of how the clinical trial data should be standardized and how it should be submitted. Now, along with along with this raw data, instead of submitting directly to the company, to the regulatory authorities, what they are doing, they are converting into one of the data model by the CDISC. CDISC has proposed a lot of data models. As a statistical programmer, you will be using two models. One is CDISC SDTM and another one is ADAM. So these are the two regulatory, these are the two models which are compatible and which are standardized, and you should submit the data only in these two forms. Now, converting the raw data into SDTM or ADAM is not possible with R. It is not possible with SAS. Sorry, it is not possible with Python. It is not even possible with Spotfire. You take Power BI, anything you take, it is not possible. Now, as a regulatory submission, who is there? Only SAS is there. But right now, R is also coming up with few packages. Few companies are contributing. Like there is a Oak package by SDTM. Oak package is there by GSK, and Roche also has given some participants some packages. We can also use them. But right now, they are in the still in the developing stage. It is not completely developed, but still it is in the trial and error method. So SAS is in the market. SAS was in the market for the last 20 years, which can convert this raw data into SDTM data sets. Now these go for submission. And what is the next one? Whatever is the data set that you have converted to SDTM data sets, you need to convert them again into ADAM data sets. Again here, who is the competitor, who is the sole person that can do that is SAS. SAS can only do that. No other tool can do that. Now, once the data sets are done, once the analysis data sets are done, then from that data only we generate tables or reports, listings, graphs, and findings, all will be generated. Now, here, converting or generating the reports, you can do it with both with SAS and also with R. Okay, they can go side by side. In industry, there will be two departments. One is SAS department, another one is R department, where the SAS department will generate their reports and R department will generate their reports. Now, these two outputs will be compared. The statistical significance, for example, R has given a p-value of 0.01 and SAS is giving us like 0.1. then you have to justify it, which one is correct. Okay, so they take a lot of care in submitting the reports to FDA. Previously, it was like, two departments will be there. Both of them are working on SAS. So a QC department will be there and production department will be there, where both the teams, they will write a code in SAS only. And the output of this department and output of this department will be compared. If both of it goes correct, then they will go for submission. If it is not, then again they have to redo it. But right now, instead of creating the reports both with SAS, as R is also very good statistical tool, they are also doing it along with the R as well. Okay, now it will be an add-on advantage for you if you also know R. Okay. So the next one is the generated tables, listings, and figures. They will go for submission in the form of a PDF documents, in the form of CSR. We call it as CSR, which is like 1000 to 2000 pages document. So this document will be submitted to FDA, or the three powerful regulatory authorities in the world which gives the market authorization is FDA from US, EMA, European Medical Agency, PMDA from Japan. So majority of the data, 80 to 90% of the global market is controlled by these three regulatory authorities. Now, for all the three regulatory authorities, the data should be submitted only through SDTM, ADAM, and TLFs. Now, who is ready to take care? Like, who is ready to miss this opportunity? So if a person knows SAS, he can convert the raw data sets and make them available for deliverables. Now, by the time that you are receiving this data, it worth almost 100 to 200 million. Even sometimes it worth more than $1 billion. Now tell me, will someone put only our software to just capture the insights? No, they will use both of them. They are not ready to play around with $1 million. The maximum amount they can spend, the maximum amount the company will be spending on analyzing or converting this raw data to FDA's submission is only $1 million to $2 million. But the value that already gained was 1 billion, and the value that are going to get is 100 billion. Now, who will play around with this small tricks? No one, right? So SAS will be there in the market and R will be there in the market. You have to learn both of them side by side. The more power you have on both of them, the better you can come up in the market. So I think this will give you some picture of what exactly happening within the clinical trial industry. And to be frank, you will be working as a statistical programmer. The role, we call it as not as a clinical SAS programmer, we call it as a statistical programmer, SP. Okay, SP means statistical programmer. So you will be working in this domain. As a SAS programmer, you will be working in converting the raw data sets to SDTM, SDTM to ADAM, ADAM to TLF, TLF to ECTD documents. Now, the people who are having 1 to 3 years of experience, they will be mostly working in converting the raw data sets to SDTM data sets. If you gain more expertise, and if it is a small project or big project also, you will be like 3 to 6 years of experience will help you in converting the SDTM datasets to ADAM datasets. And if you have more than that, or even from 3 to 10 years of expertise, you will be also preparing tables, listings, and figures. But this is just a uh, it's not confined, it's not confirmed. Okay, some companies, they may expect from you in the starting first year or second year itself to do all of these jobs. So in VRIT, what we are doing is we are not making you to learn only up to here. We are going to train you how to convert the raw data sets to SDTM data sets, and how to convert these SDTM data sets to ADAM data sets, and how to convert this to the TLFs, and how you can make it deliverable for the ECTD documents as well. So which in the first year of your entry into the SAS programming field, you will be having experience of more than 5 years of experience, because you will touch upon all the concepts and all the fields, how the system works there. So I'll go with a small demonstration. Maybe we'll take like 10 minutes to, I will demonstrate the live code. but I will be not taking any clinical trial data here. I will take small data sets because I don't want to confuse you in the starting itself. And we'll start with accessing the data. I have some data in my drive there. I'll be using that data. I will try to access that. I will show you sample code how to access, and then how to explore the data, how to prepare for our needs, how to analyze it, and then generate reports from it. And finally, I will try to export it into a PPT document, or maybe PDF, or maybe into HTML, whatever the file format we want. We will be trying to export it. Is that fine? If you have any questions so far, please go ahead. So this is a SaaS software, which is a cloud based, which I'm using, and it is free of cost, but it won't have all the tools. Okay, you may need to install the Windows software as well. So that Ravi will help you out in getting the system software. So for demonstration purpose, I'm using a cloud based software. So this is the place where we write the code and get the output. So let me show you. There is a SAS help library, and in this library you will be having a dataset called hard dataset. And this dataset is the longest ongoing clinical trial, which is FHS study, Framingham Heart Study, which started, I think, way back in 1960s or 1940s, I think. Still today, this clinical trial, this is the longest ongoing clinical trial in the world. So majority of the research findings related to the heart study is based on this study only. Like smoking causes cardiovascular disease, obesity causes cardiovascular disease, increasing in weight causes so and so thing. So female are having more cardiovascular prone disease, male are having male with obesity are prone to cardiovascular disease. Whatever the majority of the research findings, they are coming from this data only. But this data is a sample of the data. It is not the complete data. Okay, and there are like 5,200 participants who participated in this clinical trial, and the third generation clinical trial, third generation participants are participating in this clinical trial. So if you are interested more in going through that study, you can check in the Google for Framingham Heart Study. So this is the Framingham Heart Study, and the third generation participants are actually participating in it right now. You can browse it online. Okay. Now this is the sample of it. Let me use the SAS programming process. The first programming process is we access the data, then we explore data, and we prepare data. We'll try to analyze the data, and finally we'll try to export data. So let me take you through each one of these steps, but in the small manner, like small steps. I'm not going to demonstrate the complete thing because it will take 2 to 3 hours. It will take like 10-15 minutes of time to demonstrate, so that you will be comfortable of understanding what exactly going in the clinical trial sector and how the people use software tools like SAS to generate the reports. And when it comes to SAS programming language, SAS is a fourth generation programming language. So what does that mean? The fourth generation programming language, it looks like English, like how you are talking. In the similar fashion, it will be there. It won't be very syntactic. If you take C programming language, Python, or you take the low level programming languages, they will be having a lot of semicolons. There will be a lot of syntactical tools will, like the symbols will be there, and sometimes it will be difficult for you to understand the code. But in terms of SAS, whether it is as it is the fourth generation programming language, it won't take you much of the time to understand the code. It is very easy. Even to be frank, my son is only 7 years of age. He can write the basic syntax because it looks similar to English. You can easily understand it as well. So That is the first step is accessing data. Now the data is in the SAS help library. So I want to access it from the SAS help library. I want to copy it. Okay. So to do that, I'm using a data step and I'm going to create my dataset name as heart underscore copy. From where I am taking? From the SAS help library. There is a dataset name called heart and we will be taking it and run and select the code and run it. Now the copy of the hard dataset got created with the same number of observations, 5209 observations, 17 columns are there, and the data type is also preserved. The labels are preserved, so it is an exact copy. Now the copy is done. Whatever I want to do now, I can do it. I can delete some columns, I can add some columns, I can calculate some columns and all those stuff. For example, I want to analyze cholesterol here. You see in this data, we have a variable called cholesterol. Okay, this cholesterol variable is there. I want to analyze the behavior of the cholesterol in male and female participants separately. So when I want to analyze cholesterol, what it should be, the data should be sufficiently, we should have the data in that. If the data is missing, then it will be very difficult for us to analyze. Okay. Now to see how much amount of data is missing, I'll be writing a small procedure called print procedure, data equal to, now what is our data set? Heart underscore copy. And I'm requesting where if the cholesterol is missing, the cholesterol is missing, give me only that. So I'm requesting here, print the data, but wherever the cholesterol values are missing, give me only that observations. Now see, the cholesterol values are all of them are missing here. Out of those 5209 subjects, for 152 subjects, we don't have the cholesterol value. So if you see the amount of data missing is not even 10%. So it won't impact my output. So I can easily go, I can analyze it and I can present my output. I can present my insights here because the missingness is only like 5%. So 5% of missing data will not impact the 100% of the data, right? Now, the third thing is I want to calculate some summary statistics. Like what is the mean cholesterol in the whole of the participants? What is the mean cholesterol? What is the average cholesterol? These 5209 subjects have reported. Now to calculate the average, we have one more procedure called prop means. Data equal to, now what is our data set? Heart underscore copy. What is the variable I want to analyze? The column I want to analyze is cholesterol. And run. So this will help me to calculate the summary statistics. Now the average cholesterol is 227.4. What is the minimum cholesterol? There is a participant who has given, who has reported 96. There is a participant whose cholesterol is 568. So the maximum cholesterol in this whole study is, there is a person who reported 568. There are persons who reported 96 also, but average is 227.4. Now, if you ask me, sir, whether it is in male or female, so the person who reported maximum 568, is he male or female? Or if you're asking me, this 96 who has reported, is it male or female? We don't have the answer, right? Now, if you want to do it in Excel or any other tool, you have to split the data. You have to take all the female data separately and you have to calculate the summary statistics on it. And you have to take all the male data separately and calculate the average on it. But in SAS, we don't need to do that much. Just add your class variable. The class variable is gender here. Now, upon adding this statement, will give us the statistics. Now, what is the minimum in female? 117. What is the maximum in female? 493. So before we got 96 and 58, 568, both of it are reported by female itself. Sorry, by the male itself. And if you see the average is entirely different. In male, the average is 226. In female, we have slight higher average, which is 228. So this is how we explore the summary statistics. What if you want like some other statistics? Just take the same if you want to calculate the descriptive statistics, all you have to do is you have to change the name of the procedure. Instead of proc means, I'm using proc univariate procedure. And let us request. Are we able to calculate all the statistics? N, mean, standard deviation, skewness. I don't know how to calculate skewness. I don't know how to calculate kurtosis. I don't know how to calculate interquartile range. No problem. As long as you know how to use this statement and procedure, these 3 lines of code, if you know, automatically that will give you the answer. For almost 50 different type of statistics, you are able to calculate it here. What is the p-value? What is the t-value? What is the student t-test value? What is the interquartile range, standard deviation, kurtosis, all of this. Sir, I want to see how the data is distributed, whether it is normally distributed or whether it is distributed in a different manner. I want to see it. So that what we will do usually, we draw the histograms, plots, we will do that. Here, all you have to do is request for the plots. In the same procedure, if you ask plots, that will give you the plot on cholesterol. Now see here, do we have the distribution? Probability is there, box plot is there, a scatter plot is there. Now these 3 plots are automatically generated by just requesting plots. Now as a statistical programmer, you should understand what is the use of this plots keyword. Plots means it will give you the plots. Univariate, it will analyze the single variable. Data equal to, you are giving this input data set. Where means analysis variable. Run means you are executing the code. Is there any words here which are not which are very strange to us? No, right? So majority of these components are looking similar to English. So that's why we call SAS programming language is easy, even a person coming from completely non-technical background. Completely non-technical background, people can easily understand SAS. Now, apart from that, I want to prepare the data. Okay. So what we will prepare here is, I will take in every, see here we have the height and weight variables, but I don't have the BMI here. In the whole data set, we don't have the BMI. So I want to calculate BMI, the body mass index. I want to calculate it. So if you have height and weight, we can calculate it, right? We can easily calculate it. Now the third component is we have to prepare data. So we take the data step. So my output data set is going to be BMI underscore heart, where I'm going to take the heart underscore copy data set, and I want to calculate BMI here. Now what are the units of BMI? Kg per meter square, right? So kg means here the weight has the kgs, meter square means height. So can I use here weight by height? Take the weight variable and multiply it or square it by square by height, right? So this is the formula, BMI equal to weight by height. But I have a doubt here that this height is not in meters. Someone can't be 62 meters, right? This might be in inches. And this weight is not in kgs, it is in pounds. Because this data is collected from US, usually the standards are different. They will be using height in inches and weight in pounds. Now I need to convert it. To calculate the BMI, we need to convert it. So what is the conversion formula for height? So we have it in inches. So we need inches to meters. So it is giving us like almost all you have to multiply it with 0.0254. So can I add that value here in the height itself? Height is multiplied 0.0254 and weight, it is in pounds. So we need to convert it into kgs, pounds to kilos. So 0.45. Can I multiply it with 0.45 so that it will be converted? Then let us run it now. These 3 lines of code will create a new variable called BMI. For all the 5209 observations, it will be calculated. If you see, the BMI here is 24.9, 37.9, 23.7, and so on and so forth. We have a WHO classification of BMI. So I want to use that BMI classification. Let us use the BMI classification chart. So this chart shows us that if the BMI is less than 18.5, it is underweight. If a BMI is from 18.5 to 25.9, it is healthy weight. 25 to 29 is overweight. So can I categorize all of them by using this method? Let us do that. So this we call it as conditional processing the data by a condition. If that condition is true, then it will put that in that category. Okay, we'll write here if BMI is less than 18.5, then we'll create a new variable called BMI status. BMI underscore status equal to, so what is the BMI status? Underweight. Underweight. Is this easy to interpret, this statement? If BMI, it is like English, right? If BMI is less than 18.5, then BMI status equal to underweight. Is there any difficulty in that statement? It is as same as English. Now let us consider if the value is between 18.5 up to 25.9, then you have to categorize it into healthy weight. So what we do is, for sequential processing, we use else if the BMI is less than 25.9 up to 18.5, then you categorize it as healthy or healthy weight. Similarly, the same statement, because I have still some classification to be done. If it is less than 29.9, then you have to call it as overweight. So if it is 29.9, we call it as overweight. Again, for the last one, if it is more than 30, we can call it as obesity. So we can simply tell that else BMI status equal, if anything, if the value is not falling in this any of these categories, remaining all of them should go ahead with obesity. Let us run this and see the categorization is done or not. See at the last now. So 24.9 is healthy, 37.9 is obese, and then this is 26.8 is overweight, and so on and so forth. Now the categorization is done for all the 5,209 participants. Claim that our data is prepared. I want to analyze how many participants are in each category. How many participants are there in underweight? How many participants are overweight? How many participants are healthy? I want to do that. Now the fourth SAS programming process is analysis, or analyze the data. Now to analyze, I want the count of the participants, right? So we have a procedure called FREC procedure. We use that FREC data equal to, the prepared data set is BMI underscore heart, and I want the frequency table to be calculated on the BMI status variable. BMI status variable. Is this okay now? Is this giving us the output? Healthy, 3028 participants are there. Obesity, 610, 670. Overweight, 1411 participants. Underweight are 100 participants. Now we can see how the data is distributed within the BMI status. Sir, I want it in each gender also separately. Or the biostatistician, he is requesting a plot also. Some graph also he is requesting. Okay, no problem. We can request plots equal to, what type of plot you want? Sir, I want frequency plot. Run this and it will give you the frequency table, and it will also give you the frequency chart as well. Okay. Now, if you want it in each gender, this is one-way frequency analysis. What if you want to analyze two variables? How many are there in each category within each gender? Okay, then again it is complex, right? But here what we can take, we can just request a two-way frequency analysis by the gender. This will give us the output. Now, how many participants are there? 1,749 participants are female and 1,279 participants are male in the healthy category. So total are 3,028 participants. We have the row total, we have the cumulative total, we have the percentage, and every aspect is there in this total. What if you want only frequency? We don't want anything else. We have some options to control the report also. If you don't want the percentages, you can tell that I don't want the percentage, no percentage. I don't want the column percentage, I don't want the row percentage. Okay, remove all of them, give me the output table. Now the table is designed according to our wish. You don't need to copy and delete those aspects, just request it in the statement itself. So in programming language you will be learning these keywords. What are these keywords, when we use them, when we don't use them. Okay, now claim that you are you have to prepare one more report, which is the distribution of the cholesterol in each BMI status. In each BMI status, you see the same cholesterol. We did it already in using the class statement. What we did here, the cholesterol analysis, we did it by gender. But right now, what we want to analyze it by BMI status. Same procedure we write. Proc means data equal to. So this time the input data set is different because you have prepared it, right? So we'll be using that. But the variable of analysis is cholesterol. But by what class? By each BMI status class, I want to see how the cholesterol is distributed, because everyone claimed that increasing in weight increases the cholesterol, like increasing in weight increases the cholesterol, right? Let us understand here. See, who is having highest cholesterol here? People who are healthy are having highest cholesterol. So is this concept is gone now? Is there any direct relationship? You can't claim. Even someone who is overweight, they are maintaining less cholesterol than the healthy person. The person who is obese, he is having less cholesterol than this one. See the average also. Obesity by average, obesity is having 233, but healthy people are having 223. Okay, so this is the biostatistician report. The biostatistician will interpret it, what to do and how to interpret it. That is his job. Your job is to only generate the report and submit it, and he will take care of it, how to interpret and how to present it to the FDA reporters or maybe internal team discussion. That is his report, his responsibility. If you know this 4 lines, the report will come, right? Now we done with analysis. Now your client, your manager is asking it, Ashok, I want it in a PDF document and I want to present it in a seminar, so give me in the PowerPoint. So he want 2 deliverables. These 2 reports he wanted in 2 deliverables. Now usually how we create PPTs, we generate the reports in Excel, we copy it and we paste it. But in SAS we don't need to do that. We can use the ODS statement. One single statement can give you all the deliverables. So where I want to save, let me save it in, for example, in this folder. I'll copy the location of that folder. I'll tell to SAS that save the file in this folder. What should be the name? Let us take this VRIT report underscore report. And what is the extension for PowerPoint? PPTX is the extension. So what you want it in the report, you paste it there, and once it is done, close the PowerPoint. So what are the reports now you want? Already you have analyzed it, right? So your manager, he need this report. Your manager, he need this report. Your manager, he need this report as well. So paste it between those 2 ODS statements and run it. Okay, the log says that PowerPoint file is prepared. Where is it? The folder that you have given. So if you go in that folder, you have the report here. Select it, download it. If you are using the Windows version, then it will be coming up directly in your Windows laptop itself. Open the PowerPoint and establish it. That's it, done. You can change the styles, you can change the font by adding some more statement options. You can change the color of this one, you can change the font size, font color, font, the margin size, titles, footnotes. You can prepare it everything within the program itself. And you click, automatically the PowerPoint will be generated. So you have the means procedure, you have the freq procedure, and you have the frequency procedure as well. What if you have prepared it today, but tomorrow, by the time the biostatistician is going for the meeting, you have received some more data. Now, again, you don't need to start from start. All you have to do is, you have the code ready. All you have to do is change this input data set. Just change this data set. Update your data set, change the data set. Run it. Automatically, all the reports will be generated. Automatically, even the PowerPoint file will get generated. All the deliverables will be ready. Okay. So if you spend time only like, half an hour or 1 hour in the starting, whatever the challenge that comes after that, you need to change only those aspects and your program will run fine for that output delivery. Now, if the client is also asking for PDF. How can we create PDF? The same statement, but instead of PowerPoint, we will call it as PDF. I want PDF. So PDF will have an extension of PDF. And finally, we'll also tell that close the PDF. ODS, PDF, close. ODS means output delivery system. Now, when you run both of it, now the PDF file and also the PPT file both got created. Now see, we have both of them. Download it and submit it. So you have by each page, you have it here. You can do the time series analysis as well. You can create box plots, histograms, bar charts, vertical bar charts. There are huge amount of reports you can generate it with the help of SAS. For example, I'll show you one deliverable and then we will go to question and answers. So I have a data here, COVID-19 data is there, SG plot, data equal to COVID-19. So here I have the data for all the countries, COVID data for 3 years, like how many cases they have reported, how many deaths they have reported throughout this COVID pandemic. Okay, I want to plot it and I want to see it, how the trend is happening. I want to see it. Now in x-axis, I will take the date reported variable. As I'm familiar with the data set, I'm writing directly. And y-axis, I would like to add the cases. Now this will plot for us how the data got, how India has, or how a particular country has reported cases. So there are 237 countries in it. I don't want all of them. I would like to take only for India. So where country equal to India. So what I'm asking here, I'm asking a series plot where the x-axis should take the data from the date reported, y-axis should take the new cases, and I'm asking to plot the data. Now this is the trend. So this is the first wave, COVID, which happens between April and October. The peak is in October here, July and October, and the second peak was in April to July 2022, and the third peak has started in the January itself. Right? So these are the 3 peaks. Based on these 3 peaks only, we call it as first wave, second wave, third wave. The wave is because of it is going more than what it is expected or what it is previously reporting. Okay. Now, similarly, I would like to see how the trend of deaths that are reported in India. The same data set, but I'm going to change the variable. Instead of analyzing cases, we are analyzing how India has reported deaths separately. Now this is cases and this is deaths. See, sir, I don't want it for India. I would like to request for some other country. No problem. Just use the same code, change the country of your choice. If you are interested in Canada, change Canada here, and you can check it. Now this is how the Canada's report is for the new cases and for the new deaths. So it is not even crossing 400. India has crossed almost like 6,000, but COVID, see, even the cases, it is only 50,000. It is not more than 50,000. But India has crossed almost like 4 lakhs in a single day. Sir, I want to compare it both. I don't want it separate. I want to compare India and Canada in the same chart. Okay, no problem. Take the cases, same program, and instead of requesting only one country, I'm going to request both the countries, where the country should be equal to India and also Canada. So we are requesting SAS to take both of the countries and now plot the data. But we want both the data, both the countries to be separate, right? So I'm going to give one more instruction that don't consider all the data, group it by country. Keep both the countries separately. But it should be in the same plot. Now this blue line in the bottom is Canada and this red line here is India. Now it is nowhere near to India's reporting. It is not even 10% of what India has recorded. It is not even in Canada. It is not even 10%. Now we can understand what factors or what you know precautions they have taken to report very less amount of cases, and what are the factors that hugely contribute to India to report highest number of cases. You can compare it with different countries. Okay, so I'll stop here. And the deliverables, it can be sometimes they may be requesting in the form of a Word document, sometimes it will be in the PDF, PowerPoint, HTML, EPUB document, whatever it is. SAS is capable of generating them automatically. You will not do any copy paste work here. Everything is automated. Okay, so in this program, what are the components that you will be learning is you will be learning base SAS. Base SAS means not the basics of the SAS. It is a programming language within SAS itself. And you will also understand the advanced SAS, which has two main components. One is SQL, proc SQL, and another thing is macros, which will give you more power in analyzing the data. You will be understanding the CDISC fundamentals, and in that CDISC fundamentals, you will be learning what are the SDTM standards, what are the ADAM standards, and how to generate the tables, listings, and figures. And you will also, to generate the tables, listings, and figures, again we have one more tool called report writing essentials. So some specific procedures we will be learning here, like we have powerful procedures like tabulate, report, these ODS statements, and sgplot procedures. So you'll be learning them how to generate different type of tables for the deliverables, listings, figures, graphs, and those things here. So the total duration, it will take up to 80 to 100 hours, which is like 3 to 4 months or 4 to 5 months it will take roughly. And from tomorrow we will be starting the program. So whoever is interested, you can talk to Ravi and you can get your registration done. And we'll be having the sessions in the same time from 9 to 10. So I hope I make some sense to you that what exactly SAS is and how it is used in the industry, and with some insights on the clinical trial data as well. So if you have any questions related to any of these aspects, you are good to go. You can raise your questions. So our support, you will receive the all the classes will be live classes, and you will also receiving the same. If you have missed the classes, then you will be also receiving recordings for your future access, and you will also receive the notes. And every day after completion of the topic, you will be receiving the activities. Whatever the demonstrations are done, the same demonstrations and the code will also be shared with you, and the data sets for your practice, and also to accomplish the activities, we will be also sharing it with you. Okay, if you have any questions, please go ahead. I'll be here for 3 or 4 more minutes. If you have any questions, you can ask. If you don't have any questions, you can contact Ravi for your registration. So I will share the number. I think you have the access to unmute.
I'm okay with the classes, sir, but I just want to confirm the timings because I live in USA. It's already for me. It's already 10:30 in the night. If it
Okay.
starts from 9 to 10, it will take. But now it's only half an hour distance, half an hour difference. But in the morning and evening, I can manage. But up
Oh,
from next month to after that month, it will be 1.5 hour. Then it will be in the midnight for me. I'll just want to check the timings and the details of that.
okay, sure, sure.
Yeah,
We
I
can
can.
adjust it
I'll
accordingly.
yeah, I'll talk to Ravi Garu. I'll just talk to him, and I'll just confirm my details. Okay.
Sure, sure.
Yeah. Thank you very much. Have a good day, sir.